My name is Amy DuRoss. I'm the CEO of Vineti. So the topic this morning is technology trends in cell and gene therapy. For the purposes of this discussion, we're really going to talk about two major definitions of technology. So there's technology on the scientific, medical side, the underlying mechanisms in this field, and then there's the technology that is the enabling technology that's required for production and delivery of these remarkable therapies. And as we know, we're in uncharted territory here in this field because of the level of complexity and interdependency of these two types of technology. And we're at a moment, an inflection point with the first two CAR-T products out, just a veritable tsunami of discovery coming at us, where if we don't figure out some novel solutions or ways in which to address the interdependencies of these types of technologies, we actually won't enjoy an industry. We won't be able to build an industry. So we have a very esteemed panel here today who I'm going to ask them in a moment to introduce themselves and their companies and their focal points within this industry, and then we'll get right into discussion. So would you like to?
Sure. So by the way, I'm not Donna Armentano. You know, if you're looking at the order here, I'm Jim Burns. Although Donna and I used to work together many years ago, so it's good to see you up here, Donna. So I'm Jim Burns. I'm CEO of Casebia Therapeutics. And Casebia is a fairly new company. It's 2 years old. I was a third employee hired back in October of 2016. We're at about 60 employees now. We're a joint venture between Bayer and CRISPR Therapeutics. It's a very different model, which maybe I'll get into in a little bit. But I came to Casebia via Sanofi and Sanofi Genzyme. And I had been at Genzyme for a number of years. And then we were acquired by Sanofi. And I was head of what we called the North America R&D hub for Sanofi.
Great. Thanks,
Yeah,
Bob.
that's great. I'm Bob Deans. I'm Chief Technology Officer at BlueRock Therapeutics, a cousin company with Casebia, in the context that our initial financing was drawn from the same sponsors, Versant as a venture capital firm and Bayer as a pharmaceutical company. BlueRock is a cell therapeutics company in the regenerative medicine space. It's based on core developmental biology driven by producing. Our intention is to produce allogeneic therapeutic cells derived from iPSC, but we really have a second core platform technology, and that's around gene editing to enable allogeneic use and gene editing to enhance potency and produce very specific therapeutic products. I've been at BlueRock about a year. I think BlueRock, as I mentioned, is a very early new company.
Okay, James.
I'm also not Donna. I'm James Noble. I'm the CEO of Adaptimmune, which is a company that's been going a long time, which I founded quite a number of years ago. We're entirely focused on high affinity T-cell receptors, and I'm pleased to say that we announced about a month ago that we had very good responses in a second solid tumor. So we're all about solid tumors and T-cell receptors.
Donna Armentano. I'm currently at Pfizer. I've started about 2 years ago, and I head Pfizer's external science and innovation team for gene therapy. And we're focused on looking at assets and technologies to bring in that are pipeline assets, as well as technologies that can improve manufacturing, which is actually a topic that we're going to touch upon today. Prior to Pfizer, with Jim, I was at Genzyme. I was there for 24 years, and as a whole, I've had 30-plus years in developing gene therapies for rare disease.
Great, thanks. Donna, we're going to start in reverse order since you do pop up first here on the screen, and I'd love for you to just share a technology trend of high importance to you and why it's so significant.
Sure. Well, technology is important. As I was saying, there are pipeline assets, but then there's also technology that enables the movement of that pipeline. So the technology spans, I would say, many different areas or many different buckets. Manufacturing, obviously, is one of them. So some of you may recall that about 2 years ago, Pfizer acquired a company called Bamboo Therapeutics. And part of that acquisition was strategic in the sense that that acquisition brought not only the pipeline assets, but it also enabled Pfizer to jumpstart its GMP manufacturing capabilities. So with that acquisition, we became GMP phase 1/2 ready. However, it doesn't stop there. You have to think in terms of how you're going to be not just GMP phase 1/2 ready, but how you're going to be able to supply drug product and at the commercial scale. And so more recently, some of you may have heard that we've just built a new facility in North Carolina in the Kit Creek area, and that will be Pfizer's commercial manufacturing facility. But other technologies to advance the pipeline include novel capsids. We're looking for the next best technology to be able to, on many fronts, be able to reduce the dose of vector that you deliver to a patient. Obviously, if you're trying to target a liver, you want the vector to go to the liver and not elsewhere. If you're trying to target muscle, you want to go to the muscle and try not go elsewhere. But I think we heard Kathy mention this morning. I think another technological advance that's required to move the pipeline is this concept of being able to repeat dose patient. So if you think about most of the clinical trials, they've been focused on areas where delivery is to a site that's somewhat immune privileged, to the eye, to the CNS. But there are many devastating diseases in children where you know that if you deliver it once, at some point in the future, you're going to have to deliver it again. And so I think technologies that impact the immune system, immune modulation, are going to be very important for moving the pipeline into disease areas that affect infants or affect young children, where it's projected that you're going to have to repeat dose at some point.
Great. Thanks. James, please.
Yeah, thanks. As an interesting perspective, we come from the opposite end of the spectrum. We have already given multiple doses to various patients. It's not an issue with our technology. I think the thing that's going on in the technology versus the science is the fact that you're trying to create something that has an effect on patient. At the same time, you're trying to work out what the product is and how to improve it. So we've taken a very stepwise approach in terms of the most important thing for us is to get a signal, and that's why the responses in solid tumors have been so important to us. Until you do that, you can't work out how to improve the product. So at the same time, we're trying to reduce the cost of goods in manufacturing, make it more efficient. But we're also trying to look at how you improve the product. And in plain terms, that means increasing the depth and durability of the response. Now, you can do that through repeat dosing. We have repeat dosed quite a number of patients. But actually, what we're looking at is two different things. I've just come back from AACR, and anybody who's been there will have noticed the slight importance of checkpoint inhibitors. So obviously, we're looking at combination studies there. And we have one study going on in myeloma right now. We'll be initiating more in the years to come. And the second thing is to put something else into the cell that you're putting back into the patient, which what we call second generation or third generation. And these are things either designed to fight the suppressive tumor microenvironment or to induce epitope spreading, or both. And so we're going stepwise. We have generation one, you get a signal, then you work out what needs to be improved, and then you improve it. And I have to say that 3 years ago, I don't think anybody ever picked up the phone to us. And I'd say that 3 times a week, and indeed twice this morning, people have come to us with new technologies that can help do some of these things. And I think that's the challenge, funnily enough, is how on earth do you deal with the enormous number of technologies which are approaching from different angles, whether it's gene editing or whether it's any other form of vector technology or cell manufacturing technology or cryopreservation. All these things are coming at the same time, and I think that's one of the challenges for us.
It's a challenge, but it's also a pleasure having these technologies on the table in front of us. It's really the ability to go to a buffet and build the menu du jour. So I think the high-impact technology for BlueRock is really around immune engineering and synthetic biology. And I say that in 2 dimensions. First, I pointed to the fact that we are producing allogeneic cell products, and as a consequence of that, we have to ensure that they can engraft and persist. So we're really taking 2 paths to do that. One is immune editing the HLA locus and also engineering receptors to prevent innate cell clearance. We're doing that in partnership with Universal Cells. I think they've been mentioned earlier in the meeting, and we've announced a partnership with them. I'm very happy to say we've been able to, and have in our development pipeline, gene-edited, GMP-compliant master cell banks that produce the dopaminergic neurons and produce the cardiomyocytes that we're developing for our 2 therapeutic areas. I think the second dimension here is really around synthetic biology and being dedicated to receptor design and cell targeting or specific surface receptor targets. And so we see quite a bit of movement in the immuno-oncology space around tumor target selection, where people are able to build synthetic receptor complexes that apply very elementary Boolean logic, act if you see this but don't see that. So I think we see that as being very promising in terms of targeting and delivery systems going forward. And it's not lost to us, it's not lost that all of the work in effector cell development biology being used in immuno-oncology has a counterpart in autoimmune disease and tolerance. So I think we're all going to see some very creative things coming out of the T-cell space that will be applicable to cell therapeutic products that we're developing.
Great, thanks.
So for us, people ask me a lot, what keeps you awake? So Casebia is in the genetic engineering, gene editing space, and the science is so new. It's so exciting. But it's really, I think, also the technology that's going to determine whether this is going to be successful. So the way I like to think about science and technology, and I'm trained as an engineer, so we like to think of scientists as trying to understand what's out there and putting it in here. And what an engineer does, and this is where technology comes in, it's taking that and putting it back out there into something that actually works and can make a difference. And I think one of the real keys for us, and I think we probably all would agree with this, is delivery is so important, especially in the genetic engineering, the gene editing space. You have to, if you're going to go in vivo and not only just try to edit the cell by cutting the genome, cutting out the bad gene to oversimplify it, but also then insert a good gene. There's so much that you have to do. You have to get the DNA template where you need it to get to. It has to get to the cell, has to get into the cell, it has to get into the nucleus. You have the RNA that's going to guide the nuclease to where it needs to go. You need the message RNA to get to where it needs to get to. Delivery is at the heart of all of this. And for what we're trying to do now in terms of the diseases we're trying to approach, I think it's doable, but as we think about what's going to be happening next, I think, has to be a lot more invested in delivery, both viral vectors as well as non-viral. So that's, as I started, people will ask me what keeps you up at night. It's delivery, delivery, delivery. And manufacturing also, which we've already talked about, is obviously key as well. And I think we're in the gene editing space, we're lucky, as Kathy High was talking about, is that we're drafting off of 20 years of work in gene editing and ex vivo cell therapy. So we're hopeful, and I think this will be the case. It's not going to take as long to get to our first product, because we can draft off of what they've already have done for us in terms of delivery as well as manufacturing. Having said that, both of those areas, I think, are still very, very key for our success.
So picking up on manufacturing, as we have technologists, investors in the room, there are a host of these novel approaches coming forward, but if you had to prioritize the manufacturing challenges that you're facing today, and you're in various stages of development, as you've shared with us, where would you place the top 3 challenges? If you're asking for help, where would you start?
So
Jim, please. Yeah, we'll go in reverse order.
Yeah, actually, probably something you don't think about very often, but I think is an issue, because for so I'm going to be very parochial. It's about gene editing. There's 2 ways of thinking about this. You take cells out, you modify them, and you put them back in the patient, or you're going to deliver directly into the patient, and goes to the liver, it goes to you inject in the eye. And for the former, supply chain, I think, is an issue. To be able to modify the cells, ship the cells, and make sure that the quality is there. This is not trivial. And just ask the folks that developed Strimvelis. there's only one site in the world that was there to treat those patients. And that was a challenge. So as we think about developing these products, we also have to think about the supply chain. The other, and Kathy talked about this as well earlier, is that the AAV vector, the quality is extremely important. Do you have a couple of bands or do you have a smear? Because if you do some experiments and you wonder what's going on and you haven't done that quality check, then that can be enlightening. So getting the quality of the product is hugely important, and obviously capacity. Right now, that's of concern for us on the viral vector side as we look for partners in that space. I think as there are more and more companies and products that are getting into the clinic, then I'm hoping then that capacity issue will eventually be relieved because there'll be the demand for it, and suppliers will then be scaling up.
Great, thanks.
Yeah, very good. I guess for manufacturing, I worry a lot about COGS as far as staying up at night. And I think the immediate opportunity for us in terms of reducing COGS in a cell therapeutic where you're playing on developmental biology is to remove the growth factors in your expansion media. And that can be done by interrogating the genome, by epigenetic and profiling tools, and understanding the pathways that you can modulate with a small molecule alternative, or where you can use a forced transcription factor expression to drive fate of the cells instead of using an exogenous growth factor. But those are kind of the obvious places to go to drop cost of goods. I think a more important consideration is to go back and consider delivery of the final product. And so I think we all know that in delivering cells by endocardial catheter into the heart, you take a tremendous loss on cells that are expelled from the beating muscle and go back into the circulation. So that loss can vary between 90% of your cells lost, 50% of your cells lost. So an immediate way to focus on reducing effective cost of goods is to improve and understand and develop delivery systems that retain cells with high viability at the target tissue.
James, please.
So I always start with a slightly sort of controversial part about manufacturing, and that's that it's actually the type of product that comes out of it is critical to the efficacy in the field of T-cell receptors. It's, you have to have something in a solid tumor that persists for a long time, and we have persistence for about 4 or 5 years already because a solid tumor keeps having to be knocked down. So the first thing I would say about the manufacturing is the product that comes out of it has to be the right product. And we focused on that much more than the other things to start with, because if you don't get a signal, it doesn't matter how cheap the thing is, it's not going to be used. So that's the first thing. And I think the second thing is that you have to, with our T-cell technology, we have to get the viral vectors, just mentioned, we have to get that right, and we have to get the cell manufacturing right. And I think they're two completely different things. I'm certain in my own mind that the viral vector challenges can be overcome. I mean, when we first started, you might get half a dozen patients out of a $500,000 batch out of an academic institution in the USA, and now we already have worked out how to use a suspension culture, which means you can scale up the bioreactors. So it's going to become much more like a sort of monoclonal antibody. You could use small vessels, then bigger vessels, then bigger vessels. And I think that, and we've set up a partnership in the UK where we have a specific facility dedicated to ourselves just to do that, because I think relying on third parties for that is just not the way to go. I think the cell manufacturing is completely different. The cell manufacturing, frankly, is an art at the moment, and it needs to become not an art. It needs to become something which is heavily automated. It needs to become heavily processed. I think going back to the logistics, that's also important, but the key to that is you must be able to freeze the cells which you take out of the patient, and you must be able to freeze the product. That's not what Dendreon managed with Provenge, and it was unbelievable. It was impossible, I would say, to scale out. So those are the things I would concentrate on, making sure the product works, and biology is full of surprises, and you don't really know if it works till you've been in man, so that's the first thing. Then have separate strategies for vector and for the cell manufacturing, because they're trying to address different issues.
So I think at least what I worry about sometimes, and I also, this is one of the things I look for in potential partnerships, are ways to improve AAV manufacturing. So for those of you that are familiar with it, you'll know that not every vector produces with the same efficiency. So obviously anything you can do to improve yields and to improve the percent full particles that come out of that manufacturing process will impact automatically your cost of goods. Speaking of cost of goods, actually the cost of raw materials to make AAV, if you're using triple transfection, can be exorbitant if you're thinking in terms of delivering a very high dose of AAV in the context, let's say, of Duchenne muscular dystrophy. So, we look for ways to drive down the costs of those raw materials. And then, the other aspect to this is scale-up manufacturing. And it's one thing to manufacture vectors at a 50-liter scale, and it's another to do it at 200-liter scale or 2,000-liter scale. And during that scale-up process, the yields, the quality have to be maintained. So I think those are the challenges that remain in scaling up gene therapy, is to get to that 2,000-liter scale. And again, I think it speaks to the future of gene therapy and moving beyond the current disease indications when we're talking about viral gene delivery. Most of the indications are focused on rare diseases where you have small patient numbers and small doses, but how do we get beyond that? I think in order to expand into some non-rare diseases, you're going to have to meet that manufacturing challenge of getting to that 2,000-liter scale.
Mm-hmm. Let's switch topics to really focus for a moment on the business models and impact of what are potentially curative therapies and how that impacts your long-term growth and all the investment here in your pipeline. So Goldman put out a report a week ago, many of you may have seen it, really looking at the impact of these curative therapies. They took the Gilead Hep C franchise as an example, where 2015 revenues were $12.5 billion, and we're seeing a projection of $4 billion this year because essentially the technology has outpaced the prevalence of the disease. It's just the efficacy is too high. So obviously that places more pressure on the technology front, on the pipeline, on continuous iteration of the mechanisms here so that you have a really robust portfolio. And I'm so interested in your observations and how you're responding to those potential impacts. And anyone can start. We don't have to zip wire this back and forth, so please, whoever would like to jump in first.
I can maybe say something, just to allude to something that Kathy mentioned earlier this morning. So for something like hemophilia, where you already have a benchmark of the lifetime costs of treatment to a patient, so that makes your pricing model a little bit easier. It's tougher when you have diseases for which there are no previous therapies, and you're talking about death versus life, or true impact to quality of life in a patient. And it's hard to measure those things, and it's hard to put a price tag.
Go ahead, Jim.
Please,
I was going to say, I think the day when we are worried about curing cancer, we can start having these meetings and all go. The reality is you're not going to cure cancer with, in the solid tumor space. I don't think without much modification. There's going to be escape. Look at the history of the last 30 years. People thought that checkpoint inhibitors were going to cure every patient. And then guess what? And before that, monoclonal antibodies, and before that, vaccines. It has been a continuous fight, and then somehow things escape, and we have to do something else. And I just think that's going to continue in cancer. And I think the reality is just what you were saying, which is that it's the extent to which you can affect people's lives, the better, that's going to determine whether it's commercially successful. We obviously have to get the cost of goods down, but that's going to happen. You're going to be able to price these things according to what effect on the patients they have. I think in a way the payer system in the United States is not thinking about it because the patient numbers involved are not very big. And I've had that from several payers. They don't really worry because if it's a couple of thousand patients, well, it'll be smoothed out in the sort of rounding. I think it's when it gets to somebody comes up with something which is going to treat 200,000 patients a year, then I think it's going to have to be worked out much more carefully than now.
Yeah, and I think that the other part of it is as you're. First of all, I thought we're always talking about Kathy Hayek, she should be up here, but she, what she talked about in terms of a model, pricing model, and I thought that made a lot of sense. And I have to believe that if we can really cure some of these diseases that we're talking about, we'll figure out a model that makes sense, right? It makes sense so the companies earn the right to do it again. It makes sense for the payers, it makes sense for the physicians, and ultimately for the patients. But from a company standpoint, we also have to be real and think about the portfolio that we're addressing. So if you were to look at our website, you would see one of our indications is called IPEX, immune dysregulation polyendocrinopathy enteropathy X-linked. I hope you all got that. And it's a very rare disease. And we're hoping that we're going to be able to cure it, or at least make a big dent in treating that disease. And it's an entry point to other diseases that we can also impact. We can learn a lot by addressing that patient population that then allows us to also address larger populations. And so as we think about these cures and long-term treatments, you have to be thinking about the portfolio, what allows us to establish proof of concept, but then also establish it in a way where you can address a number of diseases that is not just a one-off. So it's not just the pricing model, but it's also your business model in terms of how you're thinking about the indications.
Yeah, those are really good points. So I think we're often looking in our portfolio at what we call tractable projects to carry forward. And I wasn't sure what tractable meant when we first started having the conversation. And so it means growing, or other people tell me it means growing to be a vertically integrated company where we can take a product to commercial approval and we have some credibility for marketing, performing sales and marketing. So that's instinctively not how I grew up or what I enjoy doing. I much prefer being a development engine that brings things forward to a certain point of feasibility and then doing either strong co-development or acquisition. So I think we're in a different world now where we're looking at two things that are evolving that model. One is success in the CAR T space, which has brought a tremendous amount of capital into from the investment community and the venture community into the space. So you're seeing Series A and Series B raises that are tenfold higher than they used to be. So people now have more money at their disposal to be able to execute more efficiently. The thing that's helping us along is accelerated regulatory approval. So now we're in a position with RMAT where conceivably at a phase 2B study, if not a phase 2A study, we can apply for and get market approval. So this changes the dynamic for a small company trying to build out for acquisition of an internal program, because you realize with the capital you had at the beginning, you can bring things further down the value chain. You can get more out of it when you market and sell it off. So I think it's a really healthy sign for the industry and one that we're trying to take advantage of.
Great. James, your focus has been so solidly on solid tumors, and I'm just curious, what are the technology lessons learned from liquid cancers, and how are you carrying them forward? And I'll ask that of the rest of you in general, as your observations of blood-borne cancers have evolved.
Well, I think in general, it's been wonderful to have the CAR T cells do so well in hematological tumors. I think for everybody in the room and for us in particular, I think I completely agree about the regulators. I mean, we got breakthrough status with our first program after 12 patients. And that enables you to have a whole conversation. There's no question of doing a randomized controlled study, even for a registration study. You can't do it. So we've had all sorts of help from the CAR T. So I think the lessons from us from there is that the regulators are on your side, that the approvals will come through, not just be able to do the registration study, they will actually come through, and there's tremendous value seen by big pharma companies because of that. I think in the solid tumor, the dynamics are slightly different. You need to have something that keeps going at the tumor, and the actual dynamic is that if a CAR T cell goes into and recognizes CD19, for example, it immediately sees the target. There's no tumor microenvironment. And T cells become anergic if you don't see they don't see target. And of course, they constantly do see target because B cells regenerate. So in a way, and this is not denigrating at all, it is a low-hanging fruit. It's fantastic, but it's the best way to test out the thesis, because you get immediate activation of the cells, you get constant regeneration, and then you get tremendous activity even without persistence of the cells. So I think from us, it's opened up the way to consider the way things are going to be priced. It's opened up the regulatory pathway. It's opened up tremendous interest in our technology from big pharma companies. And it's also shown us where we were going to have to differ from that. So I think for us, it's been a fantastic having people have to tread some of these paths before us. And I think we're on the next stage of solid tumors.
Anyone else like to comment, please?
I just wanted to comment on what Bob was saying, that there does seem to be a change in initial investments in companies. And I think this is really important. And the BlueRock business model and ours have something that's similar, where there was a very, very large, we'll call it a Series A. For us, it was $300 million. And the philosophy behind that is that for really new innovation, it always takes more money, more time to actually translate that into something that's going to make a difference. And so the purpose of this model is you don't get distracted by the fact that you have to go out in a couple of years and raise more money, that you have time to channel as much of your energy into doing the science, doing the development that can translate that idea into something that can make a difference. And so we're excited about trying to just help patients and do things just like the others that are working in the gene editing space, make something that's going to help patients. I'm also hoping that this model works, because this could be a new model where pharma and other investors could think about how do we translate innovation into something that really makes a difference. And so we have two reasons for us to hopefully be successful. One is for the patients. The other is that it could be a different innovation model.
Yeah, I should have said that too. I think the other thing the CAR T-cell franchise has done is shown that you can make a company worth $10 billion without any, before almost at the point if we get sales, and therefore investments scaled up. You just reminded me, because our Series A was $3 million. Not 300 million, and indeed we were running out very quickly, if I remember. And I think our second round was $2.8 million, so we didn't even make the $3 million. So that has changed. But the number of investors in meetings when I first set up Adaptimmune, it would just be the gentleman in the front row here, and we'd just chat to them because there were only about 3 people who were interested in investing. So that has changed, and that's been fantastic, obviously.
Other comments, Bob? All right. Donna, please.
I would just say that, just to reiterate some of the comments about investment, and I think one of the lessons that can be learned is just make sure that if you're going to enter into a new field with a new technology, that you give it the investment that it deserves.
Mm-hmm. Yeah, and the other point, I'm sorry, I can't resist, but it's also not just investing in what you're trying to do now. Remember what Kathy High was talking about. How long did it take for gene therapy to get to where it is? Over 20 years. Monoclonal antibodies, it took 30 years to get to where it is. So what we're doing is we're investing what we can do with what we have now, but that investment is also allowing us to invest in what's next. We're already investing in how do you in new nuclease engineering, more specificity, better delivery systems, so that we can address the problems of the future as well. So having a model that thinks about what can you do now, but also what can we do to advance the field, I think is going to poise us and others for a success that would come more rapidly than if you're just doing it incrementally like has been done in the past.
Yeah, I think to carry on Jim's point, I think the other element is increasing aptitude and intensity from the academic communities trying to out-license core technology. And so I don't know how I fall on this. I think it's tremendous to see a more mature eye on emerging technology from an institution, and the institutions are becoming much more aggressive about building business-like structures around their technology for out-licensing. I think by the same token, they're very naive about the value, and they instinctively overvalue the property that they hold on to, and it makes negotiations complex. I think to go to a point that James made earlier, we have to stack a large number of technologies to successfully build, make, and deliver our cell therapeutics, and it gets very complicated to pull technologies in that carry high licensing fees as well. So negotiations are changing.
I would say that's one of the key points of success, actually, is while it's very exciting to spend hundreds of millions on bringing in 25 different technologies, you've actually got to be linear and push things forward at the same time. And I think you need to work out very carefully what are the issues that you really need to address, and then go after the technologies that do that. And you mentioned universal cells. We did a deal, exclusive deal, actually, with all T-cell receptors with universal cells about 2 years ago, because we could see that if you needed to have an off-the-shelf product, that was one of the key things. Now we look at things like depth and durability of response, and maybe whether you can get rid of preconditioning. So there are some, I think, the danger, I think, and I've been in biotech a very long time, is the excitement of the differential technologies itself becomes an objective. And I think that's the trick, I think, the whole trick, I would say, in building a biotech company that's going to succeed and getting your commercial model right, is working out which of these things you've really got to go for and why, and then bringing them together and then pushing them in a linear fashion. If it's just spreading stuff out, I think you tend to get companies that end up with wonderful technologies and no products.
So we're asking for novel approaches to investment. This is one of the great examples, and ARM has played a seminal role in this in thinking about promulgating standards for this field ahead of any adverse events, right? So I would ask, from a technological perspective, if you could choose one topic for standardization, because we are now endowed with a standards coordinating body for this industry, what would your one item be from a technological perspective? Again, please, Donna.
When it comes to AAV, definitely standardized analytics. We've heard about the big controversy about some toxicology with patients that were delivered a certain AAV vector, and then we've heard that there were other patients that received similar doses of the same vector for which there was no tox. So I think before vectors are delivered to patients, there has to be some standardization of quality of material.
Mm-hmm. James.
Yeah, I think we have multiple areas, actually, so I think that's a very
it's hard to
difficult
choose.
one.
That's the
very difficult
yeah.
one. But I think it's trying to get, for us, I think it's trying to get a handle actually on the correlative data coming out of the patients and trying to work out which things are important. For example, I don't think anybody really knows which type of cells you need or how many you need, and going into the patient. And I think it's that understanding, so you could actually end up getting a much more standardized product to put into the patient rather than a gamish, would be very useful. So that's what we focus on.
Yeah, I have to jump all over ectopic tissue and tumor genicity testing. So in the cell therapy space, the regulators routinely require long in-life testing for ectopic tissue, abnormal tissue, or tumor genicity testing. And particularly when you're deriving products from pluripotent stem cells that have teratoma formation capability, it's almost set in stone that you go through a 9 to 12 month in-life, very expensive, $1.5 to $2 million safety study before you can file your IND and launch. So that's like driving the Indianapolis 500 and putting on the brakes and taking a break for the year, coming back and finishing the race. So it just doesn't go. So what we're seeing are the agencies waking up to using molecularly driven detection tests for tumor genicity potential, either detection of a pluripotent stem cell or looking at very specific oncogenes. But we're not there yet, and I think there has to be more of a grassroots lobby from the technology units of organizations like this to really press for shortening the development cycle.
Yeah, so I wanted to echo what Donna was saying. As you were asking me the question, that's what jumped in my head. I completely agree. The analytical side is extremely important. And we're investing quite a bit in that space. The other is, for us, is off-target effects. Mm-hmm. And how do you measure it? How do you quantify it? How do you qualify what the impact may be or may not be? Mm-hmm. And how does that carry over into it will carry over to how you design your clinical trials, right? And how you're going to have to follow patients for 15 years or however long it's going to be. So that's the other area that I think we need to have a discussion on, because you can easily see that going in different directions, and I think we have to make sure that we have the right people in the room thinking about how do we make sure we're having the right standards or approaches for understanding that.
Great, thanks. We have a few minutes left. I'd love to offer up the opportunity to ask this fantastic panel some questions. Would anyone like to jump in? I think we have a couple microphones.
And you guys are all anonymous. The lights are so bright.
Yeah, we can't see you.
We have no idea
it's like
who's
being
on the
in
other
OR.
side.
So.
Yeah, I should have put
indeed, if there is anybody.
Yeah, you're still talking to 3 people.
Anyone want to chime in? Not everyone at once. Okay, are we ready for lunch, or this is okay. We got one over here. Perfect, please.
Do you see the integration of biomaterials helping with some of
How do you see the integration of biomaterials and influencing some of the delivery? Please.
Do you mind if I jump?
Please.
I'm a biomaterials guy, so I love that question. And this is actually one of the reasons why I love the gene editing field, is that it brings together so many different parts of science and engineering to ultimately make it successful. And it would be the holy grail if you could come up with a non-viral system that could deliver all the different components that we have to deliver in gene editing. We're not there yet. I don't know if we ever will get there yet, but materials science has a ways to go before we really understand what determines the targeting to a tissue, what determines does it get into a tissue. And it's not just in humans, but it's also in our animal models, because I can deliver something to the mouse liver really, relatively easy in quotes, but that doesn't necessarily mean that it's going to get into a non-human primate or the human. And so we need to understand that, not just from a material science standpoint, but also material science plus the biology. So I think it has a big role.
Anyone else want to address that question? No? Any other questions from the... Okay, just so then... Oh, perfect, please. Go ahead, jump on in.
Okay. I'm a layman, but would you say overall progress is being made in attacking the cancer cell? It's very different. I'm no scientist, but to get into the cell, has progress really been made in the last 5 years? And is there more hope in the next 5 years?
This is a no BS question. So are we really making progress in actually the targeting of cancer cells? Whether they're liquid or they're tumor-based. Yeah.
We are really making progress. We are really making progress. I mean, 5 years ago, we had dosed a very small number of patients. We've dosed quite a lot of patients now, and we're learning a huge amount. We have now got actual data showing, solid tumor the size of a grapefruit disappearing over several months. I mean, they are enormous tumors. The metastases are dealt with by T cells. So, those two combined means that some patients are then eligible for resection because they can actually cut out what was become effectively a sort of encapsulated tumor. None of this could have happened a few years ago. And I think it's just very exciting. We're working out much more the preconditioning for the patients. We've got data that show that our T cells are right in the middle of a cold tumor. Now, a cold tumor doesn't normally, as a layman, a cold tumor sits there and it doesn't get dealt with by the immune system because there aren't any T cells to deal with it. There's nothing in the tumor. Well, there is after we've dosed patients. And it's always early steps, but you can only learn what the next step is by getting a signal. And so we know we can get our cells into the middle of a tumor. Despite all the defenses of a tumor, we know that we can get something the size of a grapefruit down to 0 over a few months. And we've just got to build on it, but I would say that's a long way from where we were 10 years ago.
So that's a great... Oh, one more. I think we have time for just one more. Please.
You're talking about getting things inside cells. There is an area of science called microbiology and delivering materials across membranes and into cells. Is there a role in this field for microbiology?
Is there room for glycobiology in
Absolutely.
this field? Yeah.
Go
ahead, Jim.
Absolutely. And I think that difference I was talking about for delivery to a mouse versus the non-human primate and the liver and the human, I think that glycobiology plays a role there. I think there's data to support that.
Okay.
Yes. So.
I think we're at the end of our time. So what you're hearing is a call to action for massive investment in this field. I think it's not BS, it's real. But we have a long way to go, so we need a lot of support from this community to get there. So thank you very much. Thanks, everybody.